Okay, so I think maybe it's the right timing to start. First of all, welcome again, everybody, and thank you for being here for this second part of our webinar series for this year. And I am glad to start back after summer vacation with our new webinar and our host, Kristy, that I will introduce shortly with her talk from the title Drug Screening with Biofabricated Tissue Models. So a few words about Kristy before we start. She's a tissue engineer at the National Center for Advancing Translational Sciences of the National Institutes of Health in the USA. And she is one of the pioneering scientists that helped establish the bioprinting protocol in the Institute. She has extensive experience in developing protocols for in vitro tissue model, especially for what concerns skin models, and how to use them to implement healthy and diseased tissue models as relevant assays for high throughput screening. So just a few technical information, and then I will stop talking. Now we will have the talk from our speaker, and after we will have a Q&A session where you can all ask any question you may have to Kristy, and I ask you kindly to do it through the chat tab or the Q&A tab of the Teams application, and I will then read them for Kristy that can provide an answer. So Kristy, it's a pleasure to have you here, and the floor is yours.
Great, wonderful. Thank you so much. Let me quickly share my screen. Hopefully it'll pop up here soon. Okay. Awesome. Can everyone see my screen clearly?
Yes, you can go forward.
Wonderful, wonderful. Well, hello, everyone, and thank you, Rajenhu, for inviting me to speak today. It is my pleasure to be sharing some of my projects and some of the amazing science that my lab has been doing. My name is Kristy Derr-Mierzwa, and I'm a research scientist and tissue engineer at the National Institutes of Health. Specifically, my center is called NCATS, which stands for the National Center for Advancing Translational Sciences. And today I will be presenting on 3D systems, drug screening with biofabricated tissue. So exactly what is translation? Translational science is the mission here at NCATS, and basically what that means is we are creating new and innovative methods and technology on the bench that will allow for more treatments to get to more patients more quickly. And we're all about bridging the gap between the science that is done on the bench and the therapeutics that are given to patients. So how do we bridge that gap? One focus is on 3D models. There are many different types of 3D models and many different types of applications for those 3D models. Some of the models include spheroids, organoids, bioprinted tissues or engineered tissues, and organ on a chip. My lab mostly focuses on spheroids, organoids, and engineered tissues. This is an extremely exciting and promising field of technology, creating more complex tissues with tailored biological components and physiological properties. These 3D models are more human-like than a 2D platform because humans are 3D. Some of the advantages of making 3D biofabricated tissues, especially when it comes to bioprinting, you can spatially control where you place different cell types, different types of extracellular matrix, and different structures. You can also control the complexity. Every assay is very context specific in science, so you can make your model as simple or as complex as your assay needs. You can also control the complexity with normal or disease cells. You can control the complexity by bioprinting physiological features such as vasculature. And lastly, the capability of doing a medium to high throughput print for drug screening. For a high throughput screen, we really focus on the principles of the 3 R's, which is replace, reduce, and refine. So one of our main goals is to reduce the amount of animal testing and research. We're using the bioprinter to create a normal tissue or non-diseased. Once we validate that healthy model, then we develop a disease model where we can infect the normal tissue with cancer cells, a virus, bacteria, etc. From there, we can screen hundreds to thousands of compounds to try to cure or reverse the disease phenotype. Within the drug discovery pipeline, we start with a 2D model to create the intended assay. Very simple, very high throughput. Then we move to screen compounds to pull out the most promising compounds for activity, where we can screen hundreds of thousands of compounds. Then we modify those hits to make them potentially suitable for humans. Next is an animal model, eventually making its way to clinical trial. So where do our 3D models fit in? Our models are going to be very valuable in the beginning stages of the drug discovery pipeline. Our 3D models can help target different drugs that are most promising in more complex models. So less than 10% of compounds entering clinical trials become approved drugs. That means 90% of the drugs in clinical trials fail. Now, there are various reasons why this happens, but one of the main reasons is that animal models that we're testing for efficacy are not biologically relevant to human. So once these compounds make it to human trials, they end up failing. The average cost to develop a drug is $2.6 billion. That's huge. So our 3D models can help mitigate not only the cost to develop a drug, but also reduce the amount of animal testing in preclinical development. So I want to quickly talk about some of the protocols we are using in our 3D bioprinting lab. So most of you may know what 3D bioprinting is. It's a process of producing a 3-dimensional tissue-like patterns of functionally viable cells in a layer-by-layer approach. Scientists can focus on organ reconstruction or regenerative medicine, or complex 3D tissue models for regenerative medicine, drug discovery and development, and then basic research or disease research. So the area we focus on is complex tissue models. We start out with a hydrogel. Most of us use a gelatin base mixed with different types of ECMs, depending on the environment or the tissue type you're working on. We take primary cells or iPSCs that are found in said tissue, mix our hydrogel and cells together to create a homogeneous mixture, load that into a syringe, place our syringe onto the bioprinter, and then print in a specific architecture that we designed on the software. Then incubate the tissue for however long the tissue needs to fully mature. We create various hydrogel scaffolds and the best scaffolding to build whatever tissue that we are intending to use. Some of the key components that we work with is a firm structure that is also able to promote cell adhesion, obviously non-toxic, And we'd like it to be native in the tissue. It needs to be printable, and then something that is also biodegradable. We would like it to be gone by the time the tissue is fully matured. So if we're using a specific scaffold within the tissue, by the time the tissue is fully matured, we would like it to be gone. So here are some of the architectures that we've been able to design using the 3D printer. So on the top corner is an example of a lumen. That's something that we could potentially print when studying vasculature. And you can see the different layers that we are able to print. The bottom corner shows a structure we print to promote vasculature growth. And this is all programmed using the software on the 3D Discovery. And some things to also consider is what type of format you may need. So I typically use transwell plates. There are 6 wells, 12 wells, 24 wells, 96 well transwells that are widely used in my lab. Or potentially if you need something more customizable. And then you also want to consider the transwell pore size and the membrane material that needs to be considered before designing your print as well. Okay, so within our lab, we have a really nice portfolio of different tissues. The skin and the retina are the original tissues when our bioprinting lab first began. These, I would say, are some of the more matured tissues out of the bunch. Our skin and retina models have a very nice reproducibility of healthy models within a variety of different disease models, such as psoriasis, melanoma, squamous cell carcinoma, atopic dermatitis. We also have a lung model, a neurovascular unit, blood-brain barrier, a neuromuscular junction. We have a placenta model and also bone, and many more. But these are our most highlighted tissues. And I'm going to be focusing on skin because those are the projects I'm working on in my area of expertise. So we published a beautiful protocol in the Tissue Engineering Part C, and this was our first full thickness tissue. And one little fact, one of our figures actually made the cover of the journal. It was their 25th year anniversary, and it was really exciting that we did make the cover. Okay, so this was a very exciting protocol because it was the first 100% bioprinted full thickness skin in a single print. This was highly automatable, reproducible, and suitable for medium to high throughput. And right now we are mostly using a 96 well transwell platform to develop our protocols. But for this publication, we did use a 12 well format, and each tissue took less than 3 minutes per tissue to print, very short incubation time, and we had a fully mature tissue in 2 weeks. We made our tissues with all vendor-provided cells, and then obviously used an open market printer, the Regenhu. And we've also validated human-like skin morphology. So you can see at the bottom the multiple layers of the stratified epidermis of the bioprinted skin equivalent compared to the human skin. We start with our hydrogel gelatin-based mixture with collagen and fibroblast. We load that into a glass syringe. The next layer is a very thin layer of laminin, and we use the microvalve needle or inkjet. And lastly, we print the keratinocytes on top, which will then proliferate and differentiate into the epidermis. So here's a screenshot of the architecture from the software that we built. We have 6 layers of dermis, so we have a relatively thick dermis, and we print the dermis thick because of contraction. lateral contraction of the dermis as the fibroblasts are moving collagen around and making collagen. So if you've ever made skin or tried to make skin, contraction is just inevitable. Then we spray a thin layer of laminin down, and this is mostly to give the keratinocytes a nice basement membrane to attach to when forming the epidermis, because the gelatin will eventually just melt away and leave the tissue. So we want to make sure that the keratinocytes, floor doesn't drop out from under them. And then lastly, we print a small spot of keratinocytes on top of the laminin layer, where they will spread out to make a monolayer and differentiate into the epidermis. So on day 7, we lower the media to air-liquid interface. So the media level will be right underneath the transwell. So here's 2 videos of what our print looks like. The first is the plunger dispense, that's the dermis. And then the other video is the keratinocytes. So just for sake of being able to visually see the print, the keratinocytes are in blue. These are normally printed in a transwell as well, but just you really wouldn't be able to see it inside the transwell. Okay. So this is what our skin looks like after printing, and then when they're raised to air-liquid interface, and then lastly, when they're fully matured, and that takes approximately 2 weeks from printing to have a fully matured tissue. One very important criteria is validating the tissue. We've gone through and completed multiple validation techniques to show our skin is as human-like as possible. The first is different immunohistochemistry stains. Every stain we've compared to real human skin tissue, which is shown below. Here's some of the ECM protein proliferation markers, different collagens, Ki67, which shows proliferating cells, the tight junction markers that are found within the epidermis, and lastly, the epidermal differentiation markers, and you can see the stain highlights the different layers of the epidermis. Next is permeability. These are very important assays to show that you have a good barrier function. We did this by doing a lucifer biotin assay. The biotin's in the red. This tests the inside out. So we put biotin underneath the tissue, and you'll see the biotin will penetrate right up to the tight junctions, but will not go past the stratum corneum, which is the very top of the tissue, the dry, flaky area that when you look down at your skin, that's what you're looking at. And then the lucifer yellow, which is shown in green, and that shows the outside in permeation. The green will not penetrate below the stratum corneum. And then off to the side, you'll see electrical impedance, which basically just measures the dryness of the surface of the skin. We've compared our full thickness skin tissue to a tissue without epidermis, and we've compared it to human skin as well. And the DPM, or dermal phase meter, is right around what normal skin measures. So you'll see that light gray bar coming across horizontally, and that's what human skin measures. And you can see our reconstructed skin tissues is right around where human skin is. And then without the epidermis, the DPMs, shoot way up very high, because the top of the skin is still wet. Another technique we used is called optical coherence tomography, or OCT, and this is a non-invasive, non-destructive imaging technique. This was nice to monitor the development of the epidermis to make sure everything looked good while the skin was maturing. And this is the image that you saw on the cover of the Tissue Engineering Journal. And lastly is the shape of the tight junctions in the epidermis, or specifically the stratum granulosum. These are called flattened Calvin's tetrachedecahedron. We used a high content microscope to image the specific shape of the cells. You can see the keratinocytes that we stained in a phalloidin to see the nice shape that creates the tight junctions. So now on to one of the first drug screens we did with our tissues. This was an awesome collaboration with Rockefeller University in New York, where we also partnered with their dermatology clinician, and this was published in Oncotarget. So the disease invasive squamous cell carcinoma is what we studied. Global incidence of SCC are 2.2 million people. So this is a pretty common skin cancer. And basically, you'll start to see the infiltration of tumor cells into the dermis of the tissue, which will then create these real nasty open sores on the exposed skin tissue, which will then penetrate deep within the dermis. Our full thickness skin samples were biofabricated that mimic normal human skin morphology in vivo. After 4 days of media submersion, the cancer spheroids were introduced that displaced and invaded the normal skin constructs. The constructs were then transferred to a custom transwell to form a construct array. The array cassettes were compatible for both incubation and tissue imaging. The tissues were treated by the addition of 1 micromolar of fluorouracil, or 5-FU, which is an extremely common chemotherapy. This was added to their nutrient-dense media underneath and then re-imaged after incubation, which was 48 hours after addition. A 3D confocal image of the skin where SCC tumor spheres were tagged with a red RFP stain, and 1% of the normal neonatal keratinocytes were tagged green. This was so we could visually image the effects of the chemotherapy and compare the ratio between the red and green before and after treatment. We did a normal H&E stain, a Ki-67, which showed proliferation of the tumor cells, and then an anti-RFP to also show the tumor growth as well. We counted the cells by fluorescent segmentation as our primary imaging biomarker before and after the 5-FU treatment period for samples fed by 5-FU treated media versus control samples fed by pure media. And for RFP SCC cells and GFP keratinocytes using different fluorescent imaging channels. So this indicated that 5-FU changed outcome for SCC versus keratinocytes. A dose response study showed increased reduction in the number of segmented cells for a 2 micromolar 5-FU treatment compared to untreated controls versus a 1 micromolar treatment compared to untreated controls. The significance of this study was remarkable. We developed a validated morphologically accurate 3D skin SCC tissue model that enables the study of the effect of chemotherapeutics on cancer cells growing in the context of their native tissue environment. 2, we developed a non-destructive 3D fluorescent confocal imaging assay to test both efficacy and general toxicity of anti-cancer chemotherapeutics. And 3, we created imaging biomarkers to quantify therapeutic effect in our system. And lastly, we pharmacologically validated our assay by demonstrating different effects of 5-FU on SCC cells versus normal keratinocytes. And wrapping things up, this is a high throughput screen we are working on, publication pending, but we did make some tweaks to this protocol. But we do print in a 96 well transwell, and in a single print, we can get around 20 plates, which we then screen over 1,000 compounds in a single drug screen with a relevant 3D model. We changed our bioprinting process a little bit to be able to print everything on top of the membrane. Instead of adding the tumor spheres to the epidermis, we print a ring of the cancer cells directly in the dermis. Basically, where you see the SCC start to form the tumors inside of the tissue, that junction between the dermis and epidermis. Here, I really want to highlight the advantages of bioprinting. So when you start to miniaturize your tissues and go down to a smaller well format, like I was explaining before, contraction, especially in skin tissue, is inevitable. So the smaller well format doesn't give you much grace when it comes to contraction. But with bioprinting, you can really control the architecture. So when you print versus pipette, when you pipette, especially in a very small well, there's nothing you can do about that meniscus that you get when pipetting. And you can see when the epidermis forms that you have that really heavy meniscus, and you don't have good differentiation in the epidermis. This is when, bioprinting becomes very crucial. So here you can see that, on the top where we bioprinted, that the epidermis formed beautifully across the tissue, with the SCC tumors inside of the dermis. Here's some validation to show where the SCC is located within the tissue. We did a Ki-67 and an anti-RFP stain to show the localization of the tumor. And then here's some confocal fluorescent imaging to show the healthy green fibroblast and the red cancer cells. Since we are imaging from the bottom, we need to tag the fibroblast better to see the green. And these results look super promising. We are really excited to continue this screen. Again, we are seeing exactly what we need to. No drug, the green and red signals stay about the same. When we test with the chemotherapy, the green stays relatively unaffected compared to the control, whereas the red goes down significantly. And then the bortezomib, which is the toxin, you can see it clearly kills most of the tissue. Yeah, these screens will be pending publication. We did a huge melanoma high throughput screen and a huge squamous cell carcinoma drug screen as well. And I would just love to thank my lab. We are an amazing team. Everyone is doing incredible projects, working on, groundbreaking research. Everyone's involved with each other's projects, and I am very lucky and blessed to work with such an amazing team. So I'd like to thank everyone at the NCATS lab, as well as all of our great collaborators that have been helping to push our research forward as well. And with that, thank you everyone so much, and I am more than happy to take any questions. I will just put myself back on.
Thank you, Kristy, for the wonderful presentation.
Thank you.
I'm really hyped about this last publication that you showed. That was a great teaser on your side.
Yes, we are so excited about it. It's going to be awesome.
And now we can start with some questions from the audience. Once again, I remind you all, if you want to just write the question in the chat tab or in the QA tab, and we will take it. I see already one about the squamous cell carcinoma experiment. How long can we keep the printed skin in the lab, and can be used to evaluate the reapparition of cancer cells? So to identify new biomarkers or potential pathways to avoid the recurrence of the cancer.
Yes, very good question. So as of right now, after our tissue reaches full maturity, I'll be honest, we don't have a super long lifespan after full maturation reaches. I would say the tissue is good for about a week after. Longer than a week, we don't want to push it too much further. We start to see that the epidermis starts to degrade somewhat. So from full maturation to about another 5 to 7 days is really the time you want to focus on, if not a little bit earlier, before that epidermis starts to degrade a bit and you no longer have a really healthy tissue.
Well, then there is the big question that we often think about. So do you see the benefit of bioprinting be good enough to really enter the pharmaceutic and cosmetic companies' practice and to overcome the easiness of 2D cell models?
Excellent question. The cosmetic industry are very interested in these 3D printed skin models. I do have a collaboration with the FDA where we are working on specific permeation assays to basically show proof of concept that our engineered skin tissue is comparable to human skin in regards to permeation. The gold standard right now is still using cadaver skin when testing cosmetics. But we are working on creating an assay that does prove that our skin is reliable when it comes to permeation. I'm pretty sure L'Oreal is using some of these reconstructed skin tissues to do some cosmetic testing on. I know in Europe, animal testing, I think, has been, banned. So this is a very hot area. So I think you'll definitely be seeing a lot more of skin tissue when it comes to cosmetic companies. I know right here in the US, we are still improving our model to get to the point where we feel comfortable that our skin is comparable to human. There's some things that we're doing with the lipid tight junction layer that needs to be improved a little bit, but I do think we're getting a lot closer. And with these 3D models, you can add more complexity to study the effects of these these cosmetics better than in 2D models.
Exactly. I think also it's one of the main points. So you really have a physiological relevance and enhanced predictiveness of the drug response thanks to this physiological approach. One example is what you've shown in terms of contraction and then formation of the layer of keratinocytes. So moving to another question, have you ever considered to implement vascularization in your tissue models, or in the case of skin, are the drugs usually administered topically?
Excellent question, and yes, we have. Vascularization is, we're starting to add it to most of our tissues that we are creating in lab. We do have vascularized full thickness skin tissue that we are also working on a pain model with nociceptors that we add vasculature to our skin model. We have our retina model has vasculature, our lung model has vasculature. So there are a lot of our models that we are adding vasculature. Again, it's very context specific. It depends on what you're looking at and what you're studying, if you need vasculature or if you don't need it. You can, there are some models that vascularization is very important in what we're studying, so it will be added to the tissue. In my case, we were more interested in studying the cancer and the tumor spheres and how they behaved in tissue, so we did not add that vasculature, but in a lot of our models, we do have vasculature. And I think you also asked drugs. And then drugs, depending on what we need to see, most of the drugs that we add to our drug screens are added underneath. But we do have some studies that we are adding the compounds on top of the tissue. It's dependent upon the compound that we're studying, whether it is added topically or if it's like a pill that you take that would be coming from underneath.
Okay, then maybe I take the opportunity to add a small question on my side. So you explained that your focus is on cancer models, and you've shown some example. I wanted just to know if you already tested these models also with other kind of diseases or conditions that could be of interest, or it's something you still have to work on in your lab?
Yeah, so within my lab, we have an HSV. So we have a herpes model that we did a beautiful, like, high throughput drug screen for the HSV herpes simplex virus. So that is one, again, publication pending. We have we're working on a psoriasis model. We have a fibrosis model. We have a paper published on atopic dermatitis as well. So, just within the skin tissue, we do have a variety of different disease models that we're working on. Just not the skin cancer, but the skin cancer, the two main drug screens that I'm focusing on right now, but other people are also have really nice projects where they're introducing high throughput drug screening.
Perfect, thank you. So another couple of questions. Could you please tell us something about the thickness of the 3D printed skin samples? And the other one is about the possibility to use these also as skin grafts and not only as in vitro models.
Okay, so for the thickness, we learned very quickly that when working with these skin cells, they're very strong. They like to make collagen. They like to move the collagen around. So contraction was a huge obstacle that we had to kind of overcome. So the way we designed our hydrogel is, it's very heavily based with fibrin. And then we polymerize that fibrin with thrombin. So that really helps with the contraction of the tissue. And also by printing the tissue right up against the walls of the transwell, so the tissue can kind of grip to the sides to help with the contraction laterally, so the tissue kind of doesn't snap like a rubber band and all come to the center. The tissue can kind of grip onto the sides of the transwell to prevent the contraction, as well as the fibrin thrombin polymerization. So, we print the tissue relatively thick, and then by the end of it, it's, probably less than half the size that we've printed it. And then what was the other?
So the other maybe are slightly connected. One is the possibility to use them as grafts. And the last one was asking the mature tissue, have you tested mechanically as a tissue arrangement, resembles an adult tissue, or could also resemble old skin or baby skin,
Yes, okay. So skin grafts, our lab has not used the skin for any type of grafts. Our lab is focused on drug screening, high throughput drug screening. We did have a very interesting collaboration with Johns Hopkins University. So they were studying amputees, and they wanted to find a better solution. So when the amputees would put on their prosthetic, it would irritate their skin. So they were trying to see if they could help these amputees make thicker skin where the prosthetic was going to be applied. So we helped Johns Hopkins University we took cells from different areas of the body. So one area we took from the bottom of the foot, and then the other area we took from the scalp. So we had thick tissue and thin tissue, and we wanted to see by taking different types of fibroblasts if it would change the thickness of the epidermis. And I think they just recently published this paper. It was so cool because depending on where you took the fibroblast, had a direct impact on the thickness of the epidermis. So in order to help these amputees, they would then take the cells from the bottom of their foot and basically inject these cells into the area where the prosthetic was going to be applied, and their skin would grow significantly thicker. So we're not specifically adding our skin as grafts to, let's say, burn victims, but we are able to create thicker skin from studying the tissue construct that we developed in lab.
Okay, that's really an exciting application.
Mm-hmm.
And then the last one is on the variability. So if it's possible to, or if it's been tested to match different kind of skin in terms of properties, different kind of skin tissue.
Okay. So this is a very interesting question as well. So we have tried taking different ages and ethnicities of keratinocytes to see how the tissue would develop. You know, we very much settled on that the younger the cells, the better and the healthier the skin made. So that's why we do lean more towards neonatal keratinocytes. The younger cells do, you know, you can, the tissue will last longer, the epidermis is thicker, it's not better differentiation. So we have used adult cells to create our skin. And again, context specific, if there's ever, you know, a disease model that may need specifically adult cells, and we have the capability of doing that. And we're also interested in learning about, you know, different ethnicities. If, you know, different ethnicities have different outcomes with penetration or, you know, different disease models as well. That's something to consider when building your tissues. And then mechanically, we personally haven't done any mechanical testing on our tissues, but, you know, that's always been a question in the back of our mind that we're interested in looking into more. So that is something that, you know, maybe in the future we'll play around with. I know, you know, for the neuromuscular junction that we've developed in-house, we've created a custom plate that has little pillars where we print our tissue around the pillars, and we're able to exercise that muscle with those pillars. So, you know, something like that could potentially help us start measuring the mechanical strength of our skin tissue.
Okay, I think we may close here. We asked you enough questions for this time. And thank you again a lot for your time and for the wonderful talk today. And I would like also to thank all the people who attended the talk, and remind you all that the recording of this talk will be available on our social channel, so you can find it there. And also please keep in tune for updates on the next event on our webinar series.
Yes, thank you so much. And everyone, if you have follow-up questions, you know, feel free to reach out via email. I'm more than happy to help with guidance and questions and whatnot. And thank you so much. I very much enjoyed this opportunity.
Thank you again, and have a nice day, you all.
Great. Thank you, everyone. Take care.